Good morning and afternoon. Today I'll be speaking to you about some of the precedents we have for making small molecule products accessible in low middle income markets and how we might apply those to the production of monoclonal antibodies for treatment and prevention in low middle income settings. Now, I want to start just by highlighting that I'm going to make a major assumption in this talk that the drug originator, who will presumably be focused on serving upper income markets, will be unable to reach the product pricing necessary to fully serve low middle income markets due to a variety of factors, both technical, but more likely related to tax and pricing law. I'm therefore going to focus on the cost of developing a biosimilar product and the economics of production of such. I will note that there are probably circumstances where this is an incorrect assumption. An example would be a small or highly unpredictable market for a product such as an Ebola treatment. And I'm happy to answer any questions about that during the question and answer section. So just to review, and I'm sure you're all familiar with this, for small molecules, typically around 30 atoms, generic small molecule products are developed as being identical to the original product in all ways in terms of the drug substance, and similar as demonstrated in bioequivalence studies in the formulation of the product. Those bioequivalence studies tend to be small and tend to be fast to run and consequently not terribly expensive. Total development cost is typically around $1 million to $1.5 million for a small molecule product. Generic manufacturers typically will use reverse engineering in the literature to identify the process to make the copy of the originator product. And there are a large number of manufacturers out there who have this skill set. So if you look at the cost driver for a small molecule, the active ingredient costs are driven by generally the cost of key starting materials, which can frequently be petroleum or fermentation based. Other raw materials required, the cost of conversion combined with the overall yield of the product. And this results in active pharmaceutical ingredient price in the range of $10,000 to $10,000 per kilo. For ARV products, as an example, we're typically seeing API prices in the $200 to $1,000 range. And this $10,000 to $10,000 per kilo price translates to a fraction of a penny to $3 for a 300 milligram product. And the cost of API will go down over time as manufacturers gain experience and reach larger scales and scale efficiency. For those small molecules, the formulation, if they are an oral product, is typically a minor driver of the product cost. Typically in a small molecule, such as an ARV, the API is in the range of 50 to 85% of the overall generic product cost, with the remainder being the formulation cost. If the product is a sterile injectable filled in individual vials, the largest cost driver is usually the sterilization, fill, and finish steps in the formulation process. So now let's look at large molecules, which includes monoclonal antibodies. These are large proteins containing thousands of atoms. The originator's product is considered a biologic. And as you all will know, there's inherent variability in biologic materials. These products are prepared from a master cell line, which is developed by the originator. And this is a major driver of cost in the development of a new product. The master cell lines used to be primarily Chinese hamster ovary, or are now more commonly humanized cells. And if we think about the idea of a biosimilar development as an analogy to generic product development, manufacturers of biosimilars, in the absence of access to that master cell line, will have to develop their own that will regenerate a similar high level of cost. And regulators are going to look for biosimilars to have no meaningful differences to the reference biologic. This essentially, in many cases, has meant in the past a requirement for new efficacy studies, which are substantially longer and more expensive than the traditional biosimilar or bioequivalent studies required for small molecules. We'll talk about that more in a moment. I want to show what a typical manufacturing process looks like for a monoclonal antibody. It typically involves several steps to take a small cell culture and scale it up to the inoculum, which will be charged into your manufacturing process. You'll continue to expand this sample in manufacturing equipment until you get to tanks of the scale we are targeting. For a large volume, high efficiency manufacturing scenario for a monoclonal antibody, we're probably looking at 20,000 liter reactors for production scale. Once the fermentation is complete, there is a long process to harvest the product and go through a work-intensive purification step involving a number of chromatography steps before we finally have our bulk product to fill. So when we look at the cost drivers associated with monoclonal antibodies, the cost drivers are substantially different than for small molecules. Because of the time intensity and the volume intensity, that is to say that we're making much less active product per liter of reactor space, the drug substance costs are driven by growth of the cell line, those multiple purification steps, very high electricity, heating, water, and reactor usage. And so this would translate in our estimate at a large scale, highly efficient, low cost manufacturer to drive costs on the order of $100,000 to $200,000 per kilo of active monoclonal antibody, or for a 300 milligram product, a market price in the range of $30 to $60. As an example, we know that Regeneron, who's developed COVID monoclonal antibody treatments, those treatments are constituted of 1.2 grams of one agent and 1.2 grams of a second agent. When you add that all together, you're looking at somewhere between $250 and $500 for the single dose product. As a comparison and reminder, the small molecule ARV costs currently for best-in-class treatment are on the order of $65 per year's worth of treatment for a 3-drug ARV regimen. So that's the drug substance. Looking at the formulation, these costs will look very similar to those for small molecule sterile fill and finish products. I will note that some products are sold as a solid in a sterile vial. That solid is prepared via lyophilization, and the time and volume intensity of that process may substantially increase the cost of fill and finish from around $2 a vial to around $10 per vial filled. Now, I want to come back and reemphasize again, if regulators require a full-scale efficacy study in order to demonstrate that the biosimilar product performs similarly to the originator, this will be a substantially expensive and prolonged process, which would drive a substantial premium on the cost of developing monoclonal antibodies. So there are a variety of things we can think about when we start thinking about how to optimize cost for monoclonal antibodies. Optimally, we would need for the biosimilar manufacturers to have a low-cost route to obtain the master cell line and the process details for growing that master cell line. So where we have a standard of voluntary licensing for small molecule products, often with included tech transfer documentation, we would additionally need access to the master cell line to have an analogous type of transfer for a monoclonal antibody. We would need to look closely at options for avoiding the need for a full efficacy study for approval. There are a variety of initiatives looking at how to do this, from regulatory procedures, which may not require running those efficacy studies, to the idea that a partnership between the originator and a potential biosimilar manufacturer to list that biosimilar manufacturer as an alternative manufacturing site in their original dossier could result in the waiving of clinical study requirements and minimize development costs. Now, on top of all of that, we need a large enough market scale to reach the economies of scale. I'd mentioned for scale for monoclonal antibodies, we're probably looking at 20,000 liter reactors. If we think about a manufacturing facility with equipment of that size and a desire at efficient scale to run it essentially year round, you're looking at around 3 million vials of product per year to be defined in our cost model as at scale. As with small molecules, there would be a particularly compelling argument to run clinical dose optimization studies to ensure that the dose required of the product is the lowest weight of active ingredient possible in order to maximize the cost efficiency of the product. Just a quick note, I think we all know that monoclonal antibodies, as a result of being proteins, are not as stable as small molecules. They're not stable in the digestive tract, and for that reason, they are by definition sterile products that are either infused or injected, and most of them require controlled temperature storage unless they're sold as a lyophilized solid. So, are there things that we can do which we've explored or have an analogy in the small molecule space to accelerate access to new monoclonal antibodies and potentially reduce their price in the market? As I mentioned, early licensing partnerships where a biosimilar manufacturer is actually engaged pre-approval and brought along as a qualified alternate manufacturing site in the original dossier could be a highly time and cost efficient approach to bringing monoclonal antibodies to low and middle income markets. But there are ways to proceed if we don't have those very early partnerships in development. As I mentioned, the time to develop these products, both because of the requirements to develop and gain experience with the master cell line, as well as getting to scale and running efficacy studies if necessary, mean that the cost of development is substantially higher than small molecules. And so financial development models where the cost of development is shared between donors and a biosimilar manufacturer should be considered. The EMA is running a pilot program for biosimilars, which would not require efficacy studies for approval. MHRA in the UK is also looking at approaches which would not require efficacy studies for approval, and innovation in the regulatory space, I think, will be critical going forward to both speeding the time to market for biosimilars and reducing the cost of development. Even if we're able to overcome all of that, we currently don't have manufacturers who have the large-scale manufacturing capability for monoclonal antibodies in low-cost settings. And so we're going to need to address the cost of installing manufacturing plants of an appropriate scale, especially if we don't want products introduced into the market where the price of those products is designed to recover the very expensive capital expenditures required for those facilities. As I mentioned, when looking at IP licensing, along with IP and tech transfer, access to master cell lines is really going to be critical to the timely development of biosimilar products and substantially reduce costs for biosimilar industry. And there's going to be a general need to invest in a multifunctional, high volume, sterile fill and finish, finished dosage form facility for a variety of products, monoclonal antibodies, but also higher volume small molecules, such as some of the newer HIV prevention products which are coming, and or other biologic manufacture. And this is going to be a separate investment requirement to the API manufacturing facility investment I had mentioned slightly earlier. I want to close just by saying that we can't forget to invest in product delivery. Everything I've spoken about assumes that a biosimilar manufacturer comes into the market with an efficient and high volume utilization of their manufacturing facility. And as I mentioned, that's roughly 3 million vials per year. If we focus on the development and regulatory approval of those products, but we are not in parallel scaling our ability to deliver those products into markets, we'll see a market failure where we have an oversupply, under procurement, and potential failure of the businesses developed to manufacture these products on a large scale. And so I just want to highlight some of the novel challenges which will complicate delivery of monoclonal antibodies, which are going to need to be addressed in parallel with an effort to install high volume, low cost manufacturing capacity. I think I'll start with a relatively minor one. As mentioned, a lot of these products will require cold chain shipping and storage. This has been addressed in the vaccine space and even in the small molecule space. It's not optimal, but this is a challenge which can be overcome. It will add marginally to the cost of products. A bigger deal is if a product has to be delivered by infusion rather than injection. Delivering infusions is not a current large-scale capability in most low and middle income public health systems. And so substantially expanded clinic space and skilled staff training, both of which are major expenses, would be required in order to develop that delivery capability. Even on an injection, subcutaneous injections are much simpler to deliver, either through self-injection or by a trained professional, as compared with an intramuscular injection. So subcutaneous delivery is substantially preferred. Products with stability and simple enough route of administration, as just mentioned, which can be delivered at the community level by community health workers with minimal training, will be substantially less expensive to deliver, and our delivery programs have substantially more reach and therefore the potential to reach more patients. And finally, the infrastructure to deliver monoclonals on a large scale needs to be built, as I mentioned, in parallel and not sequentially in order for this program to have its fastest impact. I think on that note, I will close. Thank you very much for your time, and welcome any questions during the question and answer session. Thank you.